Class 11, Chapter 6 Anatomy of Flowering Plants. You can very easily see the structure similarities and variation in the external morphology of the larger living organisms. Both plants and animals, similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organisms of higher plants. Study of internal structure of plant is called anatomy. Plants have cells as the basic unit. Cells are organized into tissues and in turn the tissues are organized into organs. Different organs in a plant show difference in their internal structure. Within angiosperm, the monocots and dicots are also seen to be anatomically different. Internal structure are also adaptation to diverse environment. 6.1 The tissue system. We were discussing types of tissue based on the types of cells present. Let us now consider how tissue vary dependent on their location in the plant body. Their structure and function would be also dependent on location. On the basis of their structure and location, there are three types of tissue systems. There are epidermal tissue system, the ground or fundamental tissue system, and the vascular or conductant tissue system. 6.1.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprise epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevent the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stomata is composed of two bean-shaped cells known as guard cells, which enclose stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of guard cells away from the stomatal pore are thin and the inner wall towards the stomatal pore are highly thickened. The guard cell possesses chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cell become specialized in their shape and size are also known as subsidiary cells. The stomatal apertures, guard cells and the surrounding subsidiary cells are together called stomatal apertures. Figure 6.1. Figure 6.1 diagrammatic representation: A stomata with bean-shaped guard cell, B stomata with dumbbell-shaped guard cell. The cells are epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.1.2 The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissue such as parenchyma, collenchyma, sclerenchyma. Parenchyma cells are usually present in cortex, pericycle, pith, and medullary rays. In the primary stems and roots in leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. 6.1.3 The vascular tissue system. The vascular tissue system consists of complex tissue, the phylum and the xylem. The xylem and phloem together constitute vascular bundles. Figure 6.2. In dicotyledonous system, cambium is present between the phloem and xylem. Such vascular bundle, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. In the monocotyledonous, the the vascular bundle have no cambium present in them. Hence, since they do not form secondary tissue, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundle usually have the phloem located only on the outer side of xylem. Figure 6.2 various type of vascular bundle: A. Radial, B. Conjoint close, C. Conjoint open. 6.2 Anatomy of dicotyledonous and monocotyledonous plant. For a better understanding of tissue organization of root, stem, and leaf, it is convenient to study the transverse section of the mature zones of these organs. 6.2.1 Dicotyledonous roots. Look at figure 6.3a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells are epiblema protrude in the form of unicellular root hair. The cortex constitute of several layers of thin-walled parenchyma cells with intercellular space. The innermost layer of the cortex, called endodermis, it comprises a single layer of barrel-shaped cells without any internal spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material serum in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cell referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth take place in these cells. The pith is small and inconspicuous. The parenchymatous cells, which are between the xylem and phloem, are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissue on the innermost side of the endodermis, such as pericycle, vascular bundle, and pith, constitute a stele. Figure 6.3 TS: A dicot root, primary, B monocot root. 6.2.2 Monocotyledous root. The anatomy of the monocotyledous root is similar to the dicot root in many aspects. Figure 6.3b. It has epidermis, cortex, endodermis, pericycle, vascular bundle, and pith as compared to the dicot root, which have fewer xylem bundles. There are usually more than 6 pluriract xylem bundles in the monocot root. Pith is large and as well as developed. Monocotyledous root do not undergo any secondary growth. 6.2.3 Dicotyledous stem. The transverse section of a typical young dicotyledous stem shows that the epidermis, the outermost protective layer of the stem. Figure 6.4a covered with a thin layer of cuticle. It may be bare trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis constitutes a few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layer below hypodermis consists of rounded thin-walled parenchyma cell with conspicuous intercellular spaces. The innermost layer of the cortex, called endodermis. The cells of the endodermis are rich in starch grains and layer is also referred to as starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of parenchyma. In between the vascular bundle, there are few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is a characteristic of dicot stem. Each vascular bundle is conjoint open with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular space with occupy the Central portion of the stem constitute the pith. 6.2.4 Monocotyledonous stem. The monocot stem has a sclerenchyma, hypodermis, a large number of scattered vascular bundle, each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Figure 6.4b. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water containing cavities are present within the vascular bundles. 6.2.5 Dorsiventral dicotyledonous leaf. The vertical section of the dorsoventral leaf through the lamina show three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which cover both the upper surface adaxial epidermis and lower surface abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The later may even lack stomata. The tissue between the upper and the lower epidermis is called mesophyll. Mesophyll, which are possesses chloroplast and carry out photosynthesis, made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval and round are loosely arranged, and spongy parenchyma is situated below the palisade cells and extend to the lower epidermis. There are numerous large spaces and air cavity between these cells. Vascular system include vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of vein. The veins vary in the thickness and the reticulate venation of the dicot leaf. The vascular bundles are surrounded by a layer of thick walled bundle sheath cells. Look at the figure 6.5a and find the position of xylem and vascular bundle. 6.2.6 Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsoventral leaf in many ways. It shows the following characteristic difference in any isobilateral leaf. If the stomata are present on both the surface of the epidermis and the mesophyll is not differentiated into palisade and spongy parenchyma, figure 6.5b. In grasses, certain adaxial epidermis cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curled inward to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar size of vascular bundle, except in many veins, as seen in the vertical section of the leaves. Summary. Anatomical. A plant is made of different kinds of tissue. The plant tissue are broadly classified into meristematic, apical, lateral, and intercalary, are permanent, simple, and complex. Assimilation of food and its storage, transportation of water, mineral, and photosynthesis and mechanical support are the main function of tissues. There are three types of tissue system: epidermal, ground, and vascular. The epidermal tissue system are made up of epidermal cells, stomata, and the epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle, and pith. The vascular tissue system is formed by the xylem and phloem on the basis of presence of cambium, located of xylem and phloem. The vascular bundles are of different types. The vascular bundles form the conductive tissue and translocate water, minerals, and food materials. Monocotyledonous and dicotyledonous plants show marked variation in their internal structure. They differ in type, number, and location of vascular bundles. The secondary growth occur in most of the dicotyledonous root and stems.